This video will show you how to use IDT's PrimerQuest tool. It's one of our free online tools to help you design PCR and qPCR assays. Before we open the PrimerQuest tool, I'm going to briefly show you two other tools used in qPCR design to make sure that you use the right tool for the best results. The pre-designed qPCR assays tool is our recommended tool to find gene expression assays for human, mouse, or rat. This tool considers the whole genome to avoid off-target effects. It provides qPCR options for multiple locations on a target, generally at exon-exon junctions. It displays and ranks optimal choices for your target from the top of the list down. The assays you select can be ordered directly from the tool. If you're interested in multiplexing two or more assays together, you'll want to use the PrimeTime Multiplex Dye Selection tool. With it, you can select appropriate fluorophores for multiplexing with your lab's instruments. Now let's get into our demonstration of the PrimerQuest tool. You'll find the tools drop-down list on the IDT homepage. Log in using your IDT username and password. To get started, enter your target sequence. There are several ways you can do this. First, you can enter it manually by cutting and pasting. Second, you can enter your NCBI accession ID for your sequence as a single ID or as a batch. Third, you can enter the target sequence on the provided Excel template form. Once you've entered your sequences, select the type of assay you want to design, standard PCR or qPCR. A primary difference between designing for standard PCR versus qPCR is the default amplicon size. In standard PCR design, it's 200 to 1,000 base pairs. For qPCR, the default amplicon size is 75 to 150 base pairs. Let's look at the design options for these two assay types. For standard PCR, your assay will have two primers. For qPCR, you have additional options. The first qPCR option is an assay with two primers for use with an intercalating dye like Cyber Green. The second qPCR option is for an assay with two primers and a PrimeTime probe, which is a fluorescently labeled 5' nuclease probe. The probe gives your assay increased specificity. Once you've selected your assay type, PrimerQuest generates designs that meet your requirements. They're ranked in sequential order with the optimal designs listed at top. By default, you'll see 5 designs per target. This can be adjusted as needed. For each assay, you'll see key information, including the target location for each oligo. To see the alignment of your oligos to your target, select View Assay Details. Further links are available for a blast search and a hairpin analysis of your sequences. We recommend performing a blast for these sequences to check for cross-reactivity on the genome of your target. We also recommend confirming acceptable dimers and secondary structure for your oligos. Something to keep in mind about the PrimerQuest tool. It designs assays for specificity within the individual targets that you've provided. It does not account for the entire genome or transcriptome of the organism. Remember, the pre-designed qPCR tool we mentioned earlier will account for the whole genome. Let's keep going. Here on this top row of buttons, if you've entered multiple targets, you can select the target you'd like to see designed assays for. If you want to order the best designs for all your targets, choose Select Top Results for all sets. Selected assays can be ordered or downloaded into Excel. At this point, use the drop-down menu to add to order. Next, select the ordering options. You can order your oligos as complete assays, mixed in one tube, or as individual oligos in separate tubes. You can also choose to receive the primers mixed together as PrimeTime primers or as single components. On this results screen, as well as on the initial target sequence entry page, you can customize your design parameters. The default parameters are IDT's general recommendations, but you can adjust them. This customization feature is one of the key strengths of the PrimerQuest tool. You can modify the criteria for the primers and probe in the assay, as well as for the target sequence. The types of parameters you can customize depend on the assay type, as well as the number of targets. As we said earlier, selecting your assay type lays the foundation for your design. So if you were to change assay type at this point, all your modified parameters would be lost. Let's walk through the most commonly modified parameters to show you how to customize your assay. You can modify GC content, melting temperature, as well as primer and probe sizes. If your targets skew towards being either AT or GC rich, you can adjust these parameters. Then the tool will optimally design an assay for your sequence. You can also incorporate predetermined parameters. Let's say you already have primers that work well and you want to design a probe that will work with them. Enter your primer sequences here. The tool will then try to design a probe in between the given primers. You can also include or exclude regions of your target to force PrimerQuest to design oligos to specific areas. For example, if you want to avoid a known SNP. You can also guide primer location on the target. Select Overlap Junction, which makes a primer overlap a specific target location. You could use this, for example, to have a primer span an exon junction. For help with any parameter in this menu, look here for definitions and more information. We hope this tutorial on the IDT PrimerQuest tool makes it easier for you to design PCR and qPCR assays before ordering them. IDT offers many other tools to help you design custom sequences. We hope you make use of those as well. Let us know if you have any questions. We are here to help you with the PrimerQuest tool or any of our online tools at application support@idtdna.com.